Okay, welcome. I think some of you probably saw me as part of the next-gen sequencing unit, and again from the bioinformatics unit that we had at the start of the year. I'm glad to see you again. I will be talking to you this week about identification of proteins by tandem mass spectrometry. Next week, I'll be right back again. We'll be talking about quantitation. I've just beefed up those slides a lot because I think there's a lot to be said about that subject. So I'm really glad to be working with you. My name is David Tabb. I'm a professor over at Stellenbosch University right across the rail yard here. And I'm really glad to get to talk to you. So I know that Ashwell and Lysex and others have also been talking about all the wonderful things that we can do with proteomics. But it's very important to me that when you use the principal algorithms that enable proteomics to be a functional science, that you don't just use them as black boxes, that you be able to look inside them and understand how they're functioning. So today we'll be talking about the algorithms that enable us to identify proteins, identify peptides really, and then infer proteins from complex samples in tandem mass spectrometry and shotgun proteomics. Okay, so we're going to start with a brief overview of tandem mass spectrometry fundamentals, because it's really, really important that you understand what those peaks represent in a tandem mass spectrum before we can really talk about how we go about identifying it. From there, we'll talk about the main workhorse of the proteomics identification pipeline, which is the database search engine. From there, we'll figure out which spectra we actually trust results from to control false discovery rates, so filtering of identifications. And finally, we'll talk about the problems of part protein assembly, of inferring a set of proteins to explain the peptides we've got, and the larger problem of parsimony in science. Okay, bioinformatics is a wildly complex space to work within. There are tools for almost every conceivable purpose within this, this amazing hierarchy of different tools. And even in the space of protein science, you can think about things like protein structure and so on and realize that there's a huge host of algorithms that surround that capacity. So in our world, we're mostly speaking about the workflows coming from tandem mass spectrometry, that's that leftmost box in the second row, feeding into database search, FDR control and protein inference, and we could also talk about post-translational modifications, a fascinating area to work in. But we're really going to just focus, in this case, on these items in this leftmost box. Now over at the right, we could also be talking about tandem mass spectrometry for metabolomics as well. A lot of that is going to be common with what we talk about next week in the space of quantitation. So this week we're going to focus on those two blue boxes there. Next week we're going to talk about the items on the right. So I think by now you probably had a few different definitions of proteomics shown to you. And maybe you have a feeling about what it represents, and maybe you don't. But I would note that there's been a fair amount of relabeling that went on as proteomics became a thing. I mean, these days, if you try to introduce a new omics, everyone kind of just gives you side-eye and says, okay, whatever. But back when proteomics was becoming an umbrella a lot of people were sheltered under, there were a lot of people who'd been working in protein science for decades before tandem mass spectrometry ever entered the field. And so a lot of them were asking the question, well, what is what I'm doing also proteomics? If I work on interactions among proteins, does that still count? And yes, the umbrella under which proteomics sits has grown quite broad. So this definition from Hoopo is not a bad declaration of what proteomics represents, in large part because it talks about all of the different different subspecialties that have come together under this space. All right. If you have spent a lot of time thinking about the bioinformatics of next-gen sequencing, you've probably spent quite a lot more time thinking about parallel measurement. For example, with a microarray experiment, you are measuring thousands and thousands of transcripts all hybridizing at the same time. When you think about whole genome shotgun sequencing, you can think about millions of different template sequences all getting sequenced at the same time in those instruments. But that is not, generally speaking, how proteomic identification experiments are carried out. For this, we need to move our thinking from parallel, parallel measurement of sequences to a serial measurement of peptides. So a lot of the advances in proteomics have not been fueled by having many, many different peptide sequences all identified at the same time, but rather by being able to identify or by be able to produce the evidence for identification of each spectrum, each tandem mass spectrum representing a peptide very, very quickly. So in milliseconds, we capture the information that causes us to recognize a particular peptide is there. Being able to capture 10,000 tandem mass spectra in the space of 90 minutes ostensibly means we have the ability to identify 10,000 peptides in the space of 90 minutes. We haven't really capitalized on that promise as much as we would like. But by being able to produce very large reams of data, by serially capturing tandem mass spectra of peptides through long experiments, has given us a lot of information from biological samples. So we can then think about the faster the tandem mass spectrometers can capture tandem mass spectra, the more sensitively we can identify the proteins of a sample. It's a shift, in any case, from parallel to sequential or series data collection. By now you should know what a mass spectrometer is. Does everyone feel really comfortable defining mass analyzer? People do not feel comfortable with that. All right. Well, let's remind ourselves that if it's not an ion, we can't measure it in mass spectrometry. So having an ion source is one of these first things that we really care about. We need to have the ability to produce ions from molecules that are floating around in solution. So the most common scenario where we use that is through liquid chromatography. So as analytes escape a column, they are electrified in a way that causes them to form this spray of ions, some of which comes into the mass spectrometer for sampling. That's electrospray ionization. John Fenn has won a lot of plaudits for his work in that space. We also have MALDI. Do people know what MALDI is useful for?
Peptide identification.
You can use it for peptide and protein identification, certainly. But generally speaking, we don't use this with with a liquid flow, but rather with samples that are dried down in spots. So you can see why it's very useful to have multiple. I'm not going to try to state the case that electrospray is better than MALDI or MALDI is better than electrospray. They serve different goals. So electrospray is very useful for us to produce ions from liquid flows of molecules, whereas MALDI is really good for dealing with dried spots of things. Okay, so those are the two ion sources we think about. I'll also talk briefly about detectors here. Detectors give us the ability to recognize that some ion has in fact passed through the mass spec. So generally we use systems like electron multiplier tubes, basically, that an ion arrives at the detector, it kicks off a couple of electrons, those electrons hit the next layer and kick off even more electrons, those electrons kick off more, and soon you have a measurable current, right? This is the basis on which a lot of our identification, a lot of our detection takes place in mass spectrometry. We also have Fourier transform-based methods for this. Across the way, we have a thermofusion mass spectrometer, and it does its detection by noticing the frequencies of currents induced by ions traveling around an electrode. So there are different ways that we end up doing detecting a signal from these mass specs. But the real business end of a mass spectrometer is in the mass analyzer. So could someone name a type of mass analyzer used in proteomics? You've had some lectures before me, right? 3, 5, 0? Am I the first speaker in proteomics? Mass analyzer known in proteomics. All right. I'm going to call my ringer. Do you have a mass analyzer for me, sir?
Orbitrap?
An Orbitrap, very good, okay. So one of the most popular mass analyzers used in proteomics today is called an Orbitrap mass analyzer. We have plenty of examples of these throughout the city, actually. The Fusion across the way is one example of an Orbitrap mass analyzer. The Q Exactives we use over at CPGR make use of Orbitrap mass analyzers. and over at UCT, the Blackburn lab uses this. So just here in Cape Town, we have multiple Orbitrap mass analyzers available to us. So an Orbitrap mass analyzer is an example of basically a spindle pole around which the ions are circulating, and the rate at which they process back and forth tells us their mass to charge ratios. That's a complex one. How about the one you have right here? We have a TOF. This is at the UWC Proteomics lab, just about 50 feet that away. We have a TOF mass analyzer that is part of a complete mass spectrometer. So a TOF is 2 time-of-flight mass analyzers. Time of flight argues that if you push a bunch of ions up to the same kinetic energy and time how long it takes them to travel a set distance, you can infer their mass to charge ratios. Okay, so I've been speaking of these as ways we can determine mass to charge ratios, and that's a large part of what mass analysis is about. But we can also do quite a few acrobatics with the ions. We can do separations, we can do isolations and things with the ions based on their mass to charge ratios. So all mass spectrometers have ion sources, mass analyzers, and detectors. The type of mass analyzer that we have frequently drives all other decisions about the mass spec. So in the case of an Orbitrap, if you may have an Orbitrap by itself, but more likely you have something like a quadrupole up front with that. So we'd call that a hybrid mass analyzer where we've got 2. The TOF is another example of a hybrid mass analyzer because it's got 2 of them. Okay, so those 3 pieces are worthwhile to know. Every mass spectrometer is going to have them. Let's talk about a broader workflow for discovery proteomics. Discovery proteomics could sometimes be called data-dependent mass spectrometry. It could be called shotgun mass spectrometry. But all of these techniques are about producing a protein inventory. What is present in the sample? So we start with a protein mixture. If I have a cell culture that I've produced from some HeLa cells, let's say, how many proteins are truly present in that mixture? Give me an order of magnitude. More or less than 100? More. Okay, so human cells require more than 100 proteins to do their thing, right? So more than 100. Is it more than 1,000? No guesses? It's more than 1,000. Okay, we got 20,000 genes. Many of them are firing off doing interesting things, right? And they have different transcripts each. Again, far more than 1,000. Generally speaking, most people would say it's well more than 10,000 proteins in these samples. So if you're starting with a mix of, conservatively counting, 10,000 proteins, and you digested it up and produced a bunch of peptides from it, you've just made the situation worse, right? You started with in excess of 10,000 proteins in the sample, you digested them with trypsin. Now each of those proteins has yielded a whole pile of peptides that make this mixture even more complex than you started with. So you started with 10,000 different proteins in there, now you've got 100,000 different peptides in this sample that you could possibly detect. One of the things that we frequently do to deal with this wild complexity of all these different peptides we might measure is called fractionation. Now this is not a method that gets used broadly enough in South Africa, but I tell you in the broader world you're going to find a lot of people using techniques like gel CMS or strong cation exchange mud pit or basic reverse phase liquid chromatography. Lots of methods that will separate that very complex mixture of proteins or peptides into simpler individual aliquots that can be analyzed independently. Okay, we use liquid chromatography. Why have I shown it as this ramp and a spike at the end? Do you know what that is? Okay, in reverse phase liquid chromatography, you have heard of reverse phase liquid chromatography in the other lectures? I'm trying to connect really hard to the things that you've heard before. You've heard at least a couple lectures, right? Have you heard the term liquid chromatography in the proteomics module? Yes, good, all right. Thank you for that. I don't want to be talking like a Martian up here, right? Okay, so in reverse phase liquid chromatography, we gradually increase the hydrophobicity of the solvent flowing through the column. This causes peptides to dislodge and start flowing out into the mass spec. People remember that much? Good, good, okay. So I've shown a typical rise in the hydrophobicity as a function of time in this liquid chromatography column. Peptides fall out of the solid phase and then go into electrospray ionization. Maybe we hit them with 3,000 volts, kapow! And these peptide ions that were already an acidic solvent suddenly take on a few extra protons. You see the peptides take on charge, both from the acidity of their environment and because of the charge we're applying to them. So now we've got this spray of charged peptide ions that we're going to measure two different ways. So this second row of things on this slide is all about two different ways that we're going to measure them. So I'm going to start over here at the left side. Now, we mentioned an Orbitrap mass analyzer a while back. This is not the only way you could do this, but typically we will find that people are using an Orbitrap or a time of flight and mass analyzer, something that gives pretty accurate masses to determine which peptides are currently visible from the stuff that's eluting from the column right now. Okay, so for each moment in time, we're going to get a snapshot of all the different peptide ions in their intact form. But we're also going to measure them a different way. Individual peptide ions that are observed in this high-resolution mass spectrum are going to be subjected to tandem mass spectrometry. So this is really three steps in one. We're going to isolate ions that are at a particular mass-to-charge ratio. I use the term mass-to-charge ratio a lot when I'm talking about proteomics, and I hope that everyone gets this concept, that an ion that has more mass to it, it will also have an associated charge, and the way that we interact with it in the mass spec is always as a mass-to-charge ratio at which we've observed some intensity, right? So we've now said we think there are a bunch of peptides of mass-to-charge 756.4 currently available in for tandem mass spectrometry. So we isolate a whole bunch of ions. We basically collect a whole bunch of ions of this sort in the trap. We cause them to collide with gas molecules, typically noble gas, so no reactions per se, but we are increasing the temperature of these peptide ions, causing them to sample a lot of conformations they wouldn't normally. And one of the outputs of this is that the peptide bonds break. Now ideally, each peptide is going to break at just one place. This one might break between the second and third amino acid, this one might break between the fourth and fifth, this one might break between the sixth and seventh. But ideally, we want each of those peptide ions to break at just one place, which produces a series of fragment ions that are indicative of protein sequence. So we started with a look at all the peptides that were there. We then focused the mass spec's attention on just one M over Z, one peptide, hopefully. We caused collision-induced dissociation, the breakage of those peptide ions into fragments, and then we measured them in a tandem mass spectrum.
Can
Yes,
I just mention it? This is where we have the parent ion and the collision, then you can get the daughter ions.
Yes, daughter ions is a term that we used a lot in the 1990s for that description. Today you often hear them called fragment ions, but I'm not sure that IUPAC prefers one term over the other at this point.
But I mentioned it to them so that
Yes.
that's it,
Yeah.
so they can just see in the picture where we give the picture.
Right. So we saw many peptides. That evidence was found in mass spectra. And then I'm going to use some poetic language. Is that okay? Each tandem mass spectrum represents the death cry of an individual peptide. We blew it apart. These are the fragments that tell its life story. All right, we're going to talk a lot more about this subject. So let's. I know that there are a lot of processes of disruption and breakage that we're discussing, and people get confused about which ones happen in the mass spec and which ones happen on the bench. So I want to try to separate this out a little bit. Denaturation and reduction. In vivo, proteins do not exist as long strings, right? They fold. They have a characteristic domain structure, etc., and they're all folded up to carry out some sort of function, because we know that the sequences of proteins leads to a particular fold leads to a particular function. Everyone remembers that. Well, I think I said structure rather than fold, but the same thing. Sequence leads to structure, structure leads to function. So we need to disrupt that in order to make these proteins more accessible to the mass spec. Denaturation is one of the things that we do. So simply soaking proteins in a bunch of urea can disrupt a lot of this folded structure that they have. What about reduction, though? What other things could prevent a protein from unfolding completely? Do proteins just hold this structure and that's just how they want to be? No, often they're kind of safety pinned into place. What's the special role of cysteine in protein structure? Cysteine is one of 2 amino acids with sulfur in its side chain. That's it, brilliant! Disulfide bonds. So the proteins don't just take on this structure and hold it out of goodwill or out of hydrophobic interactions. They're frequently safety pinned into place by disulfide bonds. So when we're causing them to stretch out through denaturation, we also want to break disulfide bridges so that they will stretch out nice and loose. And once they're all loose in this chain way, we want to use an enzyme to cleave them. So typically, we're going to use trypsin. Trypsin cuts where? Arginine and lysine, R and K, that's right. So when we have this protein unfolded in this way, once we've forced all of these disulfide bridges to be loose of each other, we can then use trypsin to cause the protein to be cut into a series of peptides. So I want to point out that points 1 and 2 do not occur in a mass spec. They occur on the bench because you are doing this to the proteins. But step 3, collision-induced dissociation, is something that happens within the tandem mass spectrometer in that vacuum environment. So the creation of fragment ions, of daughter ions, is occurring inside the mass spec. Once the mass spec has selected which peptides it wants to focus on for this series of tandem mass spectra, they're individually isolated, fragmented in the instrument to produce the fragment ions we measure in a tandem mass spec. Now, to avoid repeating myself a whole lot, I've just added in yet another slide that talks about this. That is a real mass spectrum at the upper left here. That is showing the series of, the collection of ions representing individual peptides that are present at this moment in time from what's eluting off the column. Do the same peptides elute throughout the RPLC? No, they don't. You may be running an LC separation for, say, 60 minutes, and in those 60 minutes, an individual peptide is likely only available for a closer look for about 30 seconds. Right? So we need to have a really current bit of information to decide which peptides we're going to blow apart next. So these mass spectra are generated maybe every 3 seconds. It could be a variety of durations, but let's say every 3 seconds in the course of an experiment. Now the tandem mass spectra, as I said, represent the death cries of the peptide. So we have a particular peptide that we isolated here. We remember what mass to charge value was isolated to create this tandem mass spectrum. That's what we're showing in green here. And then we blew it apart by causing the peptide to bounce off of gas molecules that caused particular peptide bonds to break and to produce these fragment ions. So a tandem mass spectrum represents the fragments of a particular peptide, generally. The mass spectrum represents the set of peptides currently visible from the column. I hope that separation is important because this is something I love writing quiz questions about. Okay, so how do we interpret a tandem mass spectrum? This is a tough one. So at the top, I have 10 amino acids, T, S, I, GTI, GPK. What relationship does trypsin have with that sequence? A quick observation. It's facile. The last amino acid is what? It's K, it's lysine. Okay, so it is reasonable to expect that the peptides we see when we've digested up a protein with trypsin will end in lysine or in arginine. This one ends in lysine, so that is not an unusual outcome. Now I've drawn in a red dotted line after the 4th amino acid there. This is to say that I am hypothesizing a breakage event that collision-induced dissociation has caused a cleavage between the second isoleucine and the first glycine, between amino acid 4 and 5 in this peptide. So how many amino acids are in the fragment to the left? 4, right. TSII are the amino acids in the fragment at the left. And how many amino acids are in the fragment at the right? 6, great. Okay, due to historic reasons, when something breaks at a peptide bond, we call the fragment that contains the left half a B ion, and we call the fragment that leaves on the right a Y ion. You can ask, where are the A, C, X, and Z ions if you like, and we can talk about it if you really want to, but it's not strictly relevant. In collision-induced dissociation, B and Y ions are the dominant ones that we see. So we call this ion TSII, including the N-terminus, the B4 ion. We call the ion on the C-terminal side that contains 6 amino acids Y6. Now I've helpfully drawn in an arrow to this tandem mass spectrum showing where those fragments appear. So we could determine what mass we expected to see for TSII. Does everyone know how? We do not. Okay. So what is the mass of serine? The residue mass of serine is 87 Daltons. What is the mass of threonine? There's a bit of shorthand on this one. If you spend a lot of time in mass spec, you start memorizing these things quick, just as a survival mechanism. So threonine is 1 methyl larger than a serine. So if you know that serine is 87, you can add 14 to that and get what? 87 and 14? Someone's got that? What is it? 101, very good, okay. So the threonine is 101, the serine is 87, isoleucine is 113, and the second isoleucine is also 113. So you can add those 4 together. In this case, add on 1 to represent the fact that it's positively charged. That's a proton sitting on it. And that gives you 415.2. 0.2. Where did that come from? That's because I rounded when I gave you the amino acid masses. That's okay. Now, we have an ion here, the B4 ion, is what happens for the first 4 amino acids. And the after ion, old joke, the Y6 ion represents the C-terminus of it. So the Y ion, we can again compute what its mass should be by adding the mass of glycine, threonine, isoleucine, glycine, proline, and lysine. We can add those together, add water, and add 1 because there's a proton sitting on this as well, and that gives us 572.36. Brilliant, right? Easy. Now, here's the difficulty. If we were to evaluate how sequence-dependent these fragments are, we would probably think to ourselves, if this breakage were not after the 4th amino acid, but were instead after the 3rd amino acid, where would the Y7 ion be? Remember, the Y6 ion appears at 572.36. How much heavier is Y7 than Y6 for this sequence? What's the only difference between their structures? Y7 moves the peptide bond cleavage 1 amino acid to the left, which means that we add to the sequence what letter? Isoleucine. Great. Okay, so isoleucine we just said has a mass of 113. So if this one, if the Y6 had a mass of 572, where would Y7's where would Y7 appear in this spectrum? 685.4. Very nice. The lady with the calculator's got it again. You're doing it on your phone? I know it's a phone. So, I mean, if you're doing it by pencil, that's even more impressive. But there you go. So 572 plus 113 is 685. So what we're seeing is that by looking at this sequence, we can interpret this spectrum. 572 is the Y6 ion, 685 corresponds to the Y7 ion, and so on. But let me push this a little bit more. It is possible to explain this spectrum if you know what sequence it represents. But would you feel comfortable interpreting this tandem mass spectrum without knowing the sequence? I took an entire semester-long course when I was in grad school on how to interpret peptide tandem mass spectra, and I tell you, I would not want to try it. This is a gorgeous spectrum. There are lots of spectrum that are quite dodgy and hard to interpret. This is not it. This is the kind of spectrum you feel very comfortable saying, oh, we know this identification really well. So you can imagine that in a dodgy tandem mass spectrum, your ability to infer sequence based on what you see in the fragments is very, very bad. Okay, so that's a starting point. I want you to understand from this slide that the fragments we see in a tandem mass spectrum are indicative of the sequence from which they come. There is a sequence dependence in which fragments we see in a tandem mass spectrum. However, we do not feel comfortable interpreting sequence from tandem mass spectra in most cases, if we don't have to do it. So, the process by which we do protein identification is going to have bench steps here at the left and computational steps at the right. On the bench, you started with a mixture of proteins. You digested that with trypsin to produce a mixture of peptides. You then ran the mass spec to collect a whole bunch of tandem mass spectra, tens of thousands of them. Now, on the flip side of that, we must undo in informatics what's been done on the bench. Having produced a bunch of tandem mass spectra, we need to put together our best hypothesis about what each tandem mass spectrum represents. We're going to transform, in other words, a collection of tandem mass spectra into a collection of PSMs, peptide-spectrum matches. Of these peptide-spectrum matches, we're going to need to apply some filters to decide which peptides we feel really confident have been identified. And having decided that this set of peptides is one that we can rely on very strongly, we will then infer a set of proteins that explain why those peptides were observed. This is the overview of the rest of the talk right here. So we need to do database search in order to get to a set of PSMs. We will use some sort of false discovery rate limitation to filter these PSMs to a set we actually trust, which is a set of peptides. And from those peptides, we need to perform inference and parsimony to decide on a set of proteins. Yes?
Can I just ask an off-topic question? When you're doing mass spec and stuff like that, obviously proteins that are like essential for life, like say now for plants Rubisco, that would now obviously show higher on mass spec. How would you like overcome that if you want to look at the proteins that's more like clearly found?
Right. The people who suffer most in that space are the people doing biofluid biomarkers. So if you were looking at plasma or serum, and you just take it neat right out of the human being, you're going to have a huge amount of albumin, and it's comparable to the Rubisco situation you're talking about with like plant leaves. So in plasma or serum, people will typically make use of what's called a depletion column to, for example, strip away 99% of the top 12 proteins always known to be found in biofluids. Depletion columns are a little, I'm not aware that they sell them for like Rubisco for particular plants, but I have to imagine that as plant proteomics grows in popularity, that will become a more popular option. More generally, I would say that this is a place where I answer with scan rate. The more rapidly we can produce tandem mass spectra of peptides, the deeper we can go into the dirt, basically. That we're not just looking at dominant peptides with really, really strong signals, but also peptides that just happen to show up for a brief moment in time at a very low concentration. So by scanning as much as is possible and ignoring stuff that we've seen in the last 30 seconds, the instrument can really sample quite deeply in sensitivity. I would say, generally speaking, at present technologies, I would say identifying more than 50,000 peptides in an individual sample is kind of what we would call high sensitivity at this point. You can get above 100,000 distinct peptides from an individual sample. Unfortunately, that's not how most people will report it in proteomics literature, though, because our field has grown very fixated on how many proteins can you detect. And in a slide I'm not going to show you, there are a large number of factors that come into play that can hugely swing the number of proteins we claim, where the number of peptides is actually much more stable. So yes, identifying thousands of proteins from a sample is certainly feasible. Identifying 100,000 different peptide sequences from samples is certainly feasible. With fractionation, not typically on 1D shots. Okay. So this diagram should be something everybody in class understands, because if you don't understand this diagram, it's going to be really hard to understand the rest of what we do in proteomics. So I really hope everyone can absorb this one well. This is a diagram of the database search engine as it was originally published in 1994. Were you guys born after that? All right, that happens. I'm old, it's okay. I was in high school when this was first published, and it was modified just 1 year later in this 1995 paper to add the all-important ability to identify post-translational modifications. So this Yates character is this perennial award winner in the mass spectrometry community. He was also my PhD advisor, so you know, so I can tell you all sorts of interesting stories about him. So let us start at the upper left. A lot of people have referred to protein identification to the process of identifying the peptide associated with a given tandem mass spectrum as looking up the answer in the back of the book, right? So we start with a FASTA database. We want to have a sequence database that shows us all of the proteins that are possible for a sample. So if you're doing clinical work, you might start with the sequence database of all proteins known for human. If you're working in wheat, you probably want to download a nice phytosome database that contains all of the proteins known for wheat. If you are working in a non-model organism, say chia, then it may be that you don't actually have a genome annotation to work from to download all of the sequences and have to work with somebody who's done RNA-seq to try to get you a list of protein sequences. But in any case, we're always going to start with a FASTA sequence database that shows all of the proteins possible in that sample. If the protein you're seeking is not in that database, you will not identify it, even if the spectra are there. This process is dependent on having a very complete sequence database to begin with. From that, the software can pull the list of protein sequences. In silico, meaning on the computer, an algorithm will digest those protein sequences to see what possible peptide sequences can result for each protein entry. So we started with protein sequences. In silico, we've turned that into a bunch of peptide sequences. Now, if we are looking for post-translational modifications like phosphorylation or methylation, I mean, there are plenty of these you could possibly go searching for, a PTM expander must then decide how can it decorate these peptides by the rules for PTMs you've laid down. So very typically, if you were looking for phosphopeptides, you would say, anytime I see a serine, I'm going to consider it at its original mass, 87 mass units, and also at its modified mass, 167 mass units. Did I get that number right? That seems wrong. It's 80 higher than 87. That should be 167, right? Okay, yeah, so this process decorates all of those peptides to consider a larger set of modified peptides to be compared. Now, all of that took place without our ever looking at the spectra, which might seem a little weird. We now need to decide which of these decorated peptides get compared to which tandem mass spectra. And we're going to work with the hypothesis here that each tandem mass spectrum represents one and only one peptide. Some new algorithms can attempt to identify multiple peptides from an individual tandem mass spectra, but I don't trust them. I'm suspicious and old. All right, so we have decorated peptides. We now need to figure out which peptides get compared to which spectra. We're going to do that on the basis of peptide mass filter. Remember that we didn't just collect a tandem mass spectrum. We also know what the peptide mass to charge value was that gave rise to this tandem mass spectrum. Generally speaking, if we're doing this with high resolution, we can tell the isotopes apart on the peptide that produced this tandem mass spectrum, which means that we know its mass very, very exactly. Very exactly. So we can rule out the comparison of peptide sequences to tandem mass spectra if the peptide mass doesn't conform very, very strongly with what we observed for that tandem mass spectrum. All right. So the things that pass this peptide mass filter are called candidate peptides. These are the different peptide sequences that we're going to compare to each tandem mass spectrum. And we're going to do that by predicting which fragments we should have seen if this sequence were present, and then compare that list of expected fragments to what we actually observed in the tandem mass spectrum. Now there's a world of database search engines published at this point. If you were trying to write a review article, write your lit review about all the different people who've written database search engines, you would come up with a very long list. We're well above 30 and probably more like 100 if we're being very, very complete of all the ones that have ever existed for this purpose. But this one was the first, back in 1994, and in the years since, the whole slew of these has been created. But almost all of them follow the same rough structure. Okay, let's talk again about the mass filter. If you are using a quadruple ion trap, this is kind of representing the old days of mass spectrometry. You might have bought an LTQ for your department or an LCQ for your department, and you would measure both peptide ion masses and their fragments in a quadruple ion trap. If you do that, you need to allow for a lot of possible error in the mass measurements that you've made of these peptide ions. Typically, allowing for an error of up to 1.25 mass to charge units in either direction. That's broad. It means that you're going to be comparing a lot of peptide sequences to each tandem mass spectrum. A lot of instruments today use time-of-flight mass analyzers, and the worst case for that is that you know the mass error within 100 ppm. But you see I changed units here, mass to charge to ppm. So this stands for parts per million. I think people recognize that. What we would do is say the observed mass of this ion was 756.4. So I divide that by 1,000,000 and then I can multiply that by 100 to say how wide is this 100 part per million mass error. More typically today, a time-of-flight mass analyzer like a triple TOF can give you mass accuracy easily within 25 parts per million. So I don't want to state that this is a hard standard everyone uses. And generally, a lot of people are using thermo-orbitrap instruments for the purposes of proteomics. In their case, they have a mass accuracy that in worst case is not more than 10 parts per million error. Generally speaking, that means something like a 0.02 m/z error for how accurate this peptide's mass is known. These things are really incredibly accurate. And having grown up with this field, I've gotten the chance to see the field move from very wide mass accuracies to very narrow ones over time. Now, how can we evaluate whether the fragments we have observed in the tandem mass spectrum are a good or bad match for the ones we would expect given this peptide sequence? Any thoughts on this one? So let's think about that peptide we were looking at a moment ago, right? It was 10 amino acids long. It might break between the first and second amino acids. It might break between the second and third. It might break between the third and the fourth. In each case, we were predicting 2 fragments to be produced, right? So you might have a B ion, you might have a Y ion. So that's a very simple case. So how many breakable peptide bonds are there in one 10 amino acid peptide? How many peptide bonds in a 10-mer? I'll wait. You look like you have an answer, right? Maybe you're just smiling. I don't know.
I tend to do that.
All right. All right, if you've got 10 amino acids and they're all connected together in a line, how many bonds do they have between them? Ah! Caught you out on that one. I heard two answers. The first answer was 10, because if you've got 10 amino acids, you've got 10 bonds. But that neglects, of course, the last one, which doesn't connect to anything, because you cleaved it with trypsin. Right? So when you have, this is called a fence post problem in bioinformatics. Here's another way to think about it. I have a fence that's 60 meters long, and I have a fence post every 10 meters. How many fence posts do I have? I don't think a lot of you have built fences in your lives. Is this what's going on? My goodness. All right. I am a farmer. I'm wearing my hat. I've got my bib overalls, everything. So, would you call me a boar at that point? Huh?
A boor.
A boor? Oh, a boor. A boor. Okay. So imagine boor tab. All right. I'm building a fence. I have a 60-meter fence, and I'm putting in a fence post every 10 meters. How many posts do I need? 5?
Why do you need every 10 meters and you have 60 meters?
It's a 60-meter fence. I'm putting in a fence post every 10 meters. How many posts do I need?
Why would you need 10?
7.
Because you're starting with one.
What's that?
7. You're starting with one.
Ah! She's got an answer for me. She says 7. Why do I need 7?
Because the one in front each end.
On each end, right. So if I'm just putting internal fence posts in, I'm going to have 5 of them, each with 10 meters of fence between them. But that still leaves me 10 meters of fence here and 10 meters of fence here. There are 4 that are bracketed and 2 that are not. So I still need to have another 2 fence posts on either side. That's called a fence post problem. Okay, so when you have a 10 amino acid peptide, we go the other way. We have 9 peptide bonds because we're only counting the boundaries between pairs of amino acids. Okay, I took a long time on that one, but thank you for hearing me out. So we have these peptide bonds. We have 9 of them. Each of them produces, we predict 2 fragment ions for each of them. So how many fragments do we predict for a 10 amino acid doubly charged peptide? 18. Very good. A B and a Y ion for each of 9 peptide bonds. Great. So our model then for what the fragments should look like will be probably to say the C-terminal ions, the Y ions, are big, and the B ions are small, let's say half the height. This is a typical kind of model. I have now predicted what the spectrum should look like, and I need to make a comparison to the tandem mass spectrum itself. The way that different search engines will score this match between expected ions and observed ions changes a lot. It is probably the most characteristic aspect of each of the search engines that have been produced. Now Sequest, as I mentioned, was published all the way back in '94. I want to point out that I've got 2 different papers here, one from 1994 and another from 2008. This is to say that 14 years after Sequest was published, the original author of the math stuff of it, Jimmy Eng, discovered a math simplification he could make that would result in this algorithm running about 10 times faster. So 14 years after this original paper was published, he said, oops, my bad, here by the way is the faster way to do it, and suddenly the same algorithm could be computed 10 times more quickly. This stuff just happens. Bioinformatics is not always about having the best answer first, but having the first answer first. Okay, so his approach is called cross-correlation. Cross-correlation. If you studied engineering at some point, this is probably something that's shown up a lot. But I want you to imagine our 18 fragment model for this peptide sequence is being compared to the spectra, this tandem mass spectrum we really collected for this peptide. And we're going to rub one past the other. You see that? All right, so we have the tau equals 0, no offset position, where everything lines up as it should. And we have slight offset values in either direction from that. And at each of these different positions, we're going to compute a score. This is a dot product score. So we're going to add together the sums of the intensity in spectrum A multiplied by the intensity aligned to it in spectrum B. So each of those values, we produce a dot product score. This is the hairball that results from that. So in this case, we have a correct sequence being compared to a particular spectrum. At tau 0, we have this huge value, and then we have a lower value that occurs when there's a slight offset. In cross-correlation, we ask how much higher is this spike in the middle of the hairball to values close to 0. And that cross-correlation score is used to rank all the different sequences in the database that could be compared to this particular spectrum. You need a computer to do that, quite obviously, because you're not just comparing each spectrum to the Tandem spectrum in an instant of time. So being able to do this very efficiently makes a very big difference. Cross-correlation scores, or XCorrs as they're frequently called, are one of the dominant ways that we can identify spectra, especially in in low signal-to-noise scenarios like ion traps. They're very good at dealing with noise. But we're not limited to just these engineering kinds of approaches. I would also talk about a jar of marbles. Imagine that our spectrum is not treated as a spectrum anymore. Now we treat it as a jar of marbles. And we have 900 white marbles in the jar to represent places where there might have been a peak, but wasn't one. We also have 100 marbles in the jar that are black. And these black marbles represent places where there could have been a peak, and there was one. Okay, that was louder, but sorry. Get sleep, everybody. All right, great. So now we have these marbles in the jar that represent locations in the spectrum, whether they do or don't have a peak, whether they're black or white marbles in the jar. Now we treat our sample as being the set of m over z values that we said should have a peak if it were this sequence. Does everyone get that? We had our 18 predicted fragment ions for this sequence. We're going to look at those 18 locations within the spectrum and draw into our hands those marbles corresponding to those locations. Now we look at our hands. Now, if I were just to draw one marble out of this jar that we described in the top one, what's the probability that it's a black marble? 1 out of 10, very nice, yeah. So we've got 1,000 marbles in the jar, 100 of them are black. So my probability that one marble drawn from the jar individually would be, is black is 10%. Great. Now, what's the probability that if I've drawn a black marble from the jar, the next one is also black? Is it the same? It's diminished a little bit, because there are now 99 black marbles in the jar and 900 white marbles in the jar. Okay, so we need some more complex math then to model this process. We've scooped out our 18 fragment ions we predicted for this sequence, and we look at our hands and we say, well, in this case I sampled out 20 marbles from this predicted peak list. 15 of them were black and 5 were white. Is that likely? Having drawn 20 marbles from that jar, is it likely 15 of them would be black? Not likely at all. But how unlikely it is really matters. So a lot of people would use the hypergeometric distribution to compute the probability that that exact outcome would take place. We had 100 black marbles in the jar, 15 of them ended up in our hands. We had 900 white marbles in the jar, 5 of them ended up in our hands. There were 1,000 marbles in the jar overall, 20 of them are in our hands. So what is the probability of that? 3.6 times 10 to the minus 12th. Well, that's a low number. That's a really low number. So this is to say that this particular outcome for this particular hypothesis about the spectrum is very unlikely to have resulted in this by chance. This is a statistical way to get at that answer. Quite different than cross-correlation was, right? Both of these are legitimate ways to go after the answer. This is something that we typically call a peak picking, a match count approach to evaluating a peptide spectrum match. We've looked at 10,000 spectra collected in this experiment. Each of them has some hypothesis put forward by the database search engine saying, this was the best sequence match I could achieve for this spectrum. Now, do you trust it or not? Typically, in a given proteomics experiment, we will accept as identified less than 50% of all the tandem mass spectra we collected. In fact, in the bad old days, it was frequently the case that you would identify less than 10% of the spectra you collected. That's kind of appalling, isn't it? But we need to figure out which of these spectra we can trust the match that was produced, and which ones we need to push aside saying, we don't know what those were. For this, we will typically use a distribution modeling route or a target-decoy route. I forget how many slides I put on this subject, not a huge number, but we're going to talk about the target-decoy route as one that's very widely used and something everybody should really understand. The distribution modeling one is more complex, but is also quite broadly used. So in these approaches, we get different outcomes, because the first approach is going to tell us whether or not to accept this particular peptide spectrum match. It's going to put a goodness score on that individual PSM. The target decoy route is quite different, because it's telling us about aggregate error. To say that we draw this threshold and only these things are above that threshold. For this set, what is the probability of error overall? What fraction of these are bogus, in other words? So these are really different outcomes from these two different approaches we use for PSM filtering. For target decoy approaches, you can think of our stack of spectra as running from my hand to the floor. Down on the floor, I have some spectra that barely matched anything. They contain maybe 10 peaks. They look like garbage. They don't identify to anything with a good score. They're just terrible. Up at the top, I have pristine, beautiful things that you would feel very comfortable putting on the cover of Nature Biotech saying, we've identified whatever it is. Okay, so immaculate beauty, awfulness down on the floor. All of the spectra fall on some continuum there. Now we need to figure out where to set a threshold across this to say, we're willing to take everything above this line in order to achieve a false discovery rate of X, maybe 5%, for example. One of the key ways that we do this is by the target decoy route. And this is incredibly widespread. In this approach, we don't just search real proteins, right? We don't just search human proteins that really are plausible. We also search for some bogus proteins. So we may, for example, use the reverse approach, where every protein in our database is matched by another protein in the database that's been reversed end for end. So we just reversed the whole sequence of the protein and said, anything that matches this we know is junk. Weird, huh? It doesn't sound like it would work, but in fact this is very, very broadly used and has held up reasonably well. So every time we apply a threshold to our big list of PSMs, we can ask how many above this threshold are from known bogus hits. And we can assume that for every one known bogus hit, there's also a hidden bogus hit in the forward sequences. So when we apply our threshold to a whole stack of PSMs, we ask how many reverse hits are above that line, and then we say an equal number of PSMs above that are also bogus but hidden among the target sequences, the non-reversed ones. Very commonly used approach. Okay, I've left out a good explanation of the Transproteomic Pipeline, of the peptide profit approach. I would note that just earlier this year, we had a Transproteomic Pipeline training session taught over at the University of Cape Town. I really would have loved to have more of you guys involved in that. But these training courses on how to use these tools are frequently available even in South Africa. So if you want to learn more about these fields, I'm happy to get you in touch with the people who program them. Okay. A really, really important point. I've spent a lot of time now talking about tandem mass spectra and peptide identifications. But what biologists generally care about are protein identifications. So when we produce tandem mass spectra in shotgun proteomics, each of those tandem mass spectra represents probably one peptide. We are going to infer a set of protein sequences that are the best explanation for the peptides that we detected. Each tandem mass spectrum has no memory of where it came from. So if you identify some 6 amino acid peptide, it probably matches to multiple proteins in the database, right? It doesn't know which one it came from. And you can't just use the fact that it appeared in minute 37 to say that it must be associated with another peptide also observed at minute 37. All these peptides were all mixed together. They don't have a way to reassociate them with the protein sequence. If you need to be very, very certain that all of these peptides come from one and only one proteoform, then you probably need to be working in top-down proteomics instead, dealing without trypsin, which is a much harder space to be. And there's a whole lot of good information about it in this Nature Methods paper. So why would a peptide appear under multiple proteins? We start with orthologs. I explained to a student of mine in another country recently that the protein sequences I have in my body are similar to the protein sequences in a blade of grass. And at first he just laughed. We talk about being related to apes, but we're of course related to all living things on this planet. So when we create sequence databases that contain multiple species, you will very frequently see that peptides from, say, a human that you've identified in your mass spec will match to sequences from a chimpanzee or to a bonobo. All sorts of possibilities. So these orthologs are one of the reasons why a sequence database can have multiple proteins that all contain the same peptides. Things like actin are really, really highly conserved among these different, among different species. On the other hand, we have a lot of genes that duplicate across the course of human evolution, which may mean that even if your database contains only human proteins, there are many different peptides that contain, many different genes that contain this same peptide. That's not the only outcome. There are also, in many of the sequence databases we work with, multiple isoforms visible for each gene, because there are multiple transcripts from each gene. If that's true, then your database will have a lot of peptides that appear among all of the different isoforms for a given gene. Anyone have an older brother or sister? I had an older brother, personally. Did any younger brothers or sisters ever get called names? Anyone want to share a name that they were called as a kid? No? We're going to leave that behind us. I can understand that. I'm going to tell you a name. My older brother, Tom, who teaches history in the middle of Missouri, if you're watching this, know that I've got my eyes on you. My older brother used to call me cheap a lot. Cheap. We would go to Walmart and we'd be looking at the toy section and I'd be, oh, I really want that Saturn V rocket, that looks really cool. And my brother would say, why don't you just buy it? And I would say, it costs too much. It costs too much. And he would say, you're so cheap! What's wrong with you? I want to point out that there's a term we apply to scientists that means something very, very similar to that. That is quite relevant. That is parsimonious. Parsimonious. In proteomics, you will frequently be faced with decisions that may cause you to claim more proteins or fewer proteins to explain a particular experiment. In science, we always try to give the simplest explanation that we can to explain our data. And so we need to be parsimonious in adding new proteins to explain the peptides that we've observed. A parsimonious answer is almost always more defensible when it comes time to meet reviewer 3 than if you try to be very expansive in your claim. I'm going to give an example. These are real data, by the way. Here we have a set of peptides shown in these little yellow bubbles that were identified on a Q-exactive instrument from a xenograft. Anyone know what a xenograft is? When we study human cancers, it's frequently very rude, not to mention unethical, to experiment on humans. I assume you've read something about that along the way? Yeah, ethics matter, right? So when we want to study a human cancer, we frequently will will grow a human tumor in a mouse, right? So you can take cells from a human tumor that you biopsied out, inject them into the mouse, and the mouse develops cancer, but it's a human cancer, a human tumor growing in the mouse organs. So that's the xenograft. So in this case, we identified a bunch of peptides, and we used a database search that included both human and mouse proteins. All of these different proteins were explanations for these particular peptides. Now, if in the bad old days of proteomics, I would have claimed all of those blue proteins had been identified. But that would not be a parsimonious approach. A parsimonious approach asks, what is the smallest number of proteins that explain all of these peptides? In this case, if we invoke this protein and this protein, we've already accounted for all of those peptides. So in this case, the parsimonious answer says just those two proteins can account for all of those peptides. We always want to seek out an answer that invokes the smallest explanation, the 2-protein answer, not the 7-protein answer, in explaining these relationships between proteins and the peptides that map to them. These can have rather large effects on the number of proteins we claim. In this case, we were shooting a sample on our instruments that had less than 50 human proteins. When we searched it against SwissProt, we saw that we had a claim of more than 400 proteins that we had identified, even though there were fewer than 50 in the sample. That's clearly not parsimonious. If we group together those proteins that are indiscernible from each other, they explain exactly the same sets of peptides, that number falls to 150 that we've claimed. That's still too much. If we apply parsimony rules on top of that, our number finally drops down to about 50, which is the true number that was in that sample. So applying parsimony and understanding problems of orthology when we use multi-species databases is a very, very big deal. Peptide identification matches tandem mass spectra scans to peptides that have been drawn from a sequence database. And those identifications, those raw identifications, require filtering to control the error rate. Everybody should get that piece. One of my favorite questions to ask is, where do the sequences come from that are matched to a particular tandem mass spectrum? We don't read the sequence from a tandem mass spectrum. That's really hard to do, and that approach requires special software called de novo sequence inference. In almost every case in proteomics, you will find that the peptide sequences that are attributed to a particular tandem mass spectrum have come through database search, which means they originated in a FASTA file. Proteins are hypotheses that explain the sets of identified peptides. As a result, when you're reading a proteomics paper, you should be really, really careful believing anyone who talks about sensitivity only by the number of proteins they identified. They're quite likely trying to get one past you. And we should be mistrustful of people, even if they're from very big, very famous labs. Okay, I hope that this is a good starter on how we go about identifying proteins. Thank you for hanging in there. I know it's a long lecture, but next week we'll be back to talk about quantification. How do we discern which proteins are different between cohorts? How do we quantify how much proteins are different among cohorts? I look forward to seeing you then.